So thank you, first of all, to the organizers for this great conference. It's been fascinating so far, and thank you for staying to this very last presentation for today. So Cellergy, well, you know, we've been talking a lot about moon shooting, so we are going, taking this technology very, very far, and we are aiming to transform human health by providing the power of young and healthy mitochondria to energize cells, fight degenerative diseases, and enhance healthy longevity. And I will tell, I mean, probably people here have some knowledge of mitochondria and some less, so I will give a little bit of background. The field of mitochondrial transplantation using mitochondria itself as the therapeutic modality is pretty new, but it is an existing field. There are a few companies out there trying to do similar things, and I'll show you exactly why we think we bring solutions where other companies could not. It is highly transformative, so you can just imagine what happens if you are able to restore energy production in cells and give the cells the tools to heal themselves, to heal every aspect of cellular function. And so the company was established 2022. We have raised so far $1.8 million, and mostly by the founders. We have now a lot of the technology in place, but in terms of preclinical, clinical data, I'll show you, we're still early, but I'll show you what we have. And we are a very experienced group of entrepreneurs working together for the past 20 years. We have developed in the past company in CRISPR gene editing, which we have sold for $300 million 4 years after inception. So from inception to exit in 4.5 years. So yeah, I've been doing a lot of complex deals, M&As, etc. So that's the team. Our gene editing company was called Amendo Biotherapeutics. So this is a team working many years together in many different ventures. And okay, so a little bit about mitochondria and why it is so powerful. So it is really mitochondria, usually called the powerhouse of the cell. It's an organelle that exists in major quantities in the cell, so it can take up to 1/4 of the cell volume. There is usually around 500 to 2,500 mitochondria per cell. Because it is energy supplying, the cell needs a lot of it. ATP, which is the energy molecule, cannot be stored, so it needs to be produced on demand all the time, wherever energy is needed. This is where the mitochondria is producing ATP. The amount of mitochondria per cell depends on the energy demand of the tissue, so more energy demanding tissues like brain, like liver, have more mitochondria per cell. And so basically what mitochondria does is take the food that we eat, the carbs, the lipids, the amino acids, and turn it into ATP. ATP is a single molecule, so it's kind of a funnel. So it takes different, we eat all kinds of different carbs, all kinds of different lipids, all kinds of different amino acids, but the mitochondria, as like sort of a funnel, takes all this food fuel and turns it into ATP, which is a single molecule energizing everything in the cell, from DNA repair, protein folding, protein trafficking, maintaining gradients across membranes, muscle contraction. Everything requires energy. And this is why this is so fundamental to every functionality of the cell. And this is also why many people believe that once energy depletes in the cell, so energy production depletes in the cell, this is where degeneration begins. So why is it depleted? What happens? With time, mitochondria accumulates a lot of damage. It is oxidized, the membrane gets oxidized, the DNA accumulates mutation. Mitochondria has its own DNA, so this is outside of the microbiome, and on the cellular level, it's quite unique. There is no other organelle in the cell that has its own DNA, and the reason is that mitochondria used to be a bacteria, so it's kind of a, it's a symbiosis. It's a bacteria became dormant in the eukaryotic cell, and this is what actually gave rise to all multicellular life on Earth. So when we started working in the field of mitochondria, we were quite amazed to find that people are injecting mitochondria IV in multiple animal models, multiple disease models for, well, probably about a decade now, and getting really phenomenal results. So when you, in Parkinson's disease, for example, even in Duchenne muscular dystrophy, which is a genetic disease, very severe one, so in a genetic model, when you inject mitochondria IV, you see complete amelioration of the disease symptoms. The same is for metabolic diseases like fatty liver disease, and even Leigh syndrome, which is, again, a genetic disease. When you inject mitochondria into these disease models, you get phenomenal results. So why is this not translated to people yet? And that was our first question. So, like, what's missing? Why is this amazing thing not yet in humans? And there are actually three answers to this. So three what we call the pillars of our technology that are missing, and we put them in place to really make mitochondria transplantation therapy a reality. So one is the source. So the source of mitochondria, and I'll go back to this slide for a second. In all these different studies, they were using mitochondria from sources that are not relevant for human use. So all of these studies use mitochondria from either transformed cell lines or animal tissues. So taking mouse liver, extracting mitochondria, and then injecting it to another mouse model or some disease. Or alternatively, they're taking mitochondria from HeLa cells or from HepG2. So these transformed cell lines that you can grow extensively in the lab and you can do experiments with, but are not translatable to human therapeutics. You cannot take mitochondria from cancer cell lines and make it a therapy to people. So source was the first thing that we needed to solve. So the source had to be non-transformed. It has to be primary cells. It has to be scalable. We still need very large amounts. And it has to be high quality in terms of the mitochondria with high mitochondrial content. And that was the first thing we put in place. And we've actually used cells that we can culture in very, very large amounts in bioreactors. Now we can make, we can scale, we're now working to scale it up to 1,000 liters of cells in bioreactors, which are primary